de la Universidad de NYU Pain Research Center en Nueva York, y posteriormente, en el 24, se mudó a la Universidad de Florida, donde actualmente es el director del Centro de como, digamos, dato cultural, tiene casi 700 artículos, unos muy buenos. Presentarles al doctor Rajesh Khanna.
Thank you very much. I'll just have a shout out to Paz, Kimberly, and Eric. They're in the audience in New York and Texas and in Florida. You can hear me, right? They just said Dorchito, so it's good. Well, thank you very much. It's an honor to be in Mexico. I love Mexican people, as you will find out very quickly, and I love everything about Mexico. It's my first trip here. I had a wonderful time in the symposium that's happening. And today I'll tell you a little bit about what the wonderful people in the lab, most of whom are Mexican, trained by these gentlemen right here, and some of the people that you know are doing in the US in my lab. So the story is going to focus only on one channel today. I talked about a couple of channels yesterday, but today's story is going to focus on a very important channel that's critical for pain research and for pain, and hopefully we can come up with something, a new drug. I just heard of a new toxin about 1.7, so very, very. Body. And that in itself is the problem of why we cannot solve chronic pain. It is, where do you start? Where do you start with such a difficult problem, right? It's a constellation of many diseases. It's thousands of diseases all sort of into one. But again, we have to start somewhere, and for today's talk, I will start in the periphery. It's been said by many famous scientists that all you need is the excitation or drive in the periphery for the
train of neuropathic pain or train of excitability to start that causes chronic pain. And this is what Dr. Raja from Johns Hopkins has said, and this is sort of cartooned in this slide, where we think that targeting these channels, which decorate the sensory neurons that you all know about, where all the wonderful channels are expressed that cause and are targeted for pain, all along the axon in the spinal cord and up into the thalamic part. So we want to target something in the periphery to stop the signal of pain where it starts. And so to get everybody on the same page, I know it's a pharmacology department, you guys know very well about these channels, but I'll begin at the beginning. Pain starts in the periphery at some sort of a stimuli is sensed, and that stimuli leads to the transmission of some action potential that's driven largely by sodium channels. And this really is happening within the confines of these wonderful neurons, these neurons called the sensory neurons, the dorsal ganglion neurons. Channels, including 1.7, 1.8, 1.9, that drive the action potentials. Not only do they drive the excitability of a neuron to convey the message of pain, the signaling of pain, but they also contribute to transmitter release through their actions on subsequent channels, so for example, the calcium channels. And collectively, they all contribute to the phenomena of nociception, or pain as we know it. But again, biology is not simple. It's always complicated. Pain is complicated. So one important channel is the one in purple, Nav1.7. It contributes to that rising phase of the action potential, without which you will not have the action potential fire. Now, we know the large part of it, the upstroke, is driven by 1.8. But clearly, 1.7 is super critical. Without 1.7, you can't have the action potential, in the most cases, be driven upwards. So think of this as the volume knob of pain. Pain. Who knows who this is?
Jimi Hendrix.
Thank you. Showing my age, These guys are like, no, I don't know who this is. Jimi Hendrix. So Jimi Hendrix is playing his guitar next to a big ass amplifier. So he's playing his guitar not with his fingers, but with his teeth. And what I'm trying to show you here is that Hendrix putting the playing the guitar with his teeth. That's 1.7. So you can stop the signal or the music of Hendrix two ways. What can you do? You can stop him here, or you just unplug the amplifier. This is 1.8. This is 1.7. So we're going to try to block 1.7. There is a new drug on the market from the FDA that targets 1.8. This part. It's called Jurnavic. So why this channel? This is a very important channel. Many, many years ago, about 20, 25 years ago, it was discovered that this channel has mutations that cause gain of function, so much more sodium coming in, which leads to lots of pain disorders, paroxysmal pain disorders. On the other side, you have mutations that cause the channel to close and not enough sodium comes in, which means that you don't have enough action potential. And this leads to congenital insensitive of pain disorders. These people have no pain. This gentleman is now about 27, 28 years old. When he was 6 months old, he chewed out his tongue. He feels no pain. And these people do not live very long because of accidents, because of all kinds of things that they do to injure themselves. So this is an important channel. You mutate it one way, it causes pain. Mutate it a different way, it doesn't cause any pain. So could be the single gene that could become the volume knob of pain. One gene to control all pain. Amazing concept, very sexy, very good to go after, right? But why not? Why not? You could make a lot of money. They could be rich and solve pain forever. Turns out, as with big grand ideas, it was a complete disaster. Unmitigated disaster. Everybody tried, everybody failed. Here's some examples. Biogen, they had some compounds that they tried in this and failed clinically. Biogen had another compound for diabetic neuropathy, absolutely failed, withdrawn during painful diabetic neuropathy. So a lot of drugs have been developed, some beautiful compounds, but they all have been garbage. Nothing works. Why? Here are some reasons why. We don't know how the channel works completely. So we're working on that. A lot of people across the globe are still studying the mechanisms of the biology of 1.7. What about other channels? It has other family members. Maybe they're affected. Maybe they're blocked. So specificity and selectivity become a very important problem. What about other subunits? And this is something I'll harp on a lot because this is what we do. Off-target effects. So even the best compound that has been developed that is ready for clinical use is being used clinically. If I give it to any one of you, what will happen is your blood pressure will drop and you will faint. So they cause autonomic dysfunction. And this is the biggest problem with these directly targeted compounds. They all, every single one of them, will engage in this unwanted side effect. Terrible. And so this is sort of the big problems that's facing the field right now. And this is something we are trying to overcome in our strategy. And of course, there's some other issues, but I won't go through them. So with that preamble, with that idea that it is a problematic to address and to target this channel, we came up with the question, is there a different way of targeting this channel? And what we did is something that is not brand new. People had thought about this idea before. For example, Hughes Abriel in Switzerland, along with De Kostrid's group, they many years ago found the idea that a protein called NET42, it's a kinase that regulates the channel itself and can affect phosphorylation and other activities of the channel that can control the activity of the channel. And they went after this idea, and I don't know if they actually developed anything from it, but biologically it was already known that the channel doesn't sit in the membrane by itself. It has a lot of partners that it talks to, and it can be regulated. So with this idea, we mined the literature and of all the proteins that could potentially interact with the 1.7 channel, and found one protein that we selected as our favorite, and this was the protein called CRIM2, CRMP2, gene name is down here. What is it? It's a cytosolic protein, it's a tetramer. Its main role, it was never identified in the context of any channels. Its main role is to collapse the axon when it's growing. So if you haven't, during development, axons grow, they find their cognate partner, and they make a synapse. So the role of this protein, upon application of a compound called somatostatin, of a ligand called somatostatin, it would collapse the growth cone. And that's what its role. And that's how it got its name, collapse in response mediator protein. So nothing had been known in the context of whether it talks to channels or what else it does. Basically, it regulates elongation and outgrowth of neurons. And my lab over the last 15 years or so has really worked on this protein with the idea that this protein could be a regulator of the trafficking of these ion channels that we're interested in, specifically the pain channels. All right, so how did we go from that idea of picking this protein out of a hat to does it have an effect on the channel? So here's a first experiment. So we envisioned that the channel interacts with CRIM2. So what if we just take out CRIM2, this auxiliary protein that we think? Well, we did that using siRNA, the protein's gone. And when we did that, you can see beautiful recordings in normal cell from normal DRGs, TTX sensitive, so that's 1.7 including. And you can see a very nice reduction. If you add back a knockdown resistant version of this CRIM2, you can fully recapitulate the currents. So CRIM2 is itself enough. If you delete it, you regulate the channel. The TTX resistant channels are not affected. The 1.8, 1.9, they're not touched with this phenomena. Okay, it's hard to delete a channel or delete a protein from a body. It's not going to happen. Nobody's going to use that as a therapy for the most part. So what if we can do something much more subtle? So we looked at the primary structure of this protein, and with my wife's group, we solved the crystal structure of CRIM2, shown down there. And we started to look at what kind of modifications are happening. And you can see there are a lot of phosphorylation modifications that happen to this protein. There's a kinase site that's very important. my lab for the first time discovered a sumolation site, SUMO, so small ubiquitin-like modifier, and it happens to occur at a lysine residue at position 374. There are two others that are there, but they're not very exposed, and by elimination and site-directed mutagenesis studies, we figured out that it was just that one amino acid, 374. So to convince you what it does, if you look at this panel on the right, if you mutate the sumolation site from CRIM2, no, remember, we're only touching CRIM2, we're not doing anything else to these cells. And add that to cells, you can see, again, a marked reduction that we saw upon complete deletion. So here, we're only mutating one amino acid of this protein. So, and you can also get this reduction if you take enzymes that cleave the SUMO, the SNP, the proteases, and they also result in reduction of the current. So we're beginning to get the idea that sumolation of CRIM2 could be a very important regulator of the sodium channels. To take this into sensory neurons, the real pain cells, we did the same experiment, but this time we separated them into resistant and then with some other manipulations to figure out whether it's 1.7 or something else. And what we're doing here is taking sensory neurons, transfecting either wild-type CRIM2 or a sumo-null CRIM2, the 374A mutant. And you can see the resistant channels are not affected. The sensitive currents, TTX-sensitive currents, are indeed suppressed, but this is composed of 111617. So how do we know it's 1.7 specifically? Well, we do that by adding in protox. Protox inhibits 1.7. When we eliminated 1.7 out of the equation, now the currents are the same. So that eliminates 1.6 and 1.1. So by inference, the regulation is all based on 1.7. And we also did 1.3, 1.1, 1.5, and none of those were affected. So it's a selective regulator of 1.7. Importantly, as we go forward, and I'm going to show you, based on this mechanism, we'll make a transgenic animal, we'll develop a drug, but it's important to de-risk your mechanism in humans. Ultimately, if this is to work, it has to work in humans, right? So we took human DRGs and did the same manipulation we did earlier in rat DRGs or mouse DRGs. We put wild-type CRIM2 and recorded. You see beautiful sodium currents in human DRGs. These are massive cells, massive currents. And upon transfection of the sumo-null mutant, you now see a massive suppression of the TTX-sensitive, but not the resistant. So again, the mechanism transcends. So that was pretty exciting. And the reason for that suppression is because of trafficking. Here's an example showing you why that result. So if you take cells, transfect them with this wild type or mutant, and you label and use confocal to look at sodium channels on the surface. In the wild type CRIM2, you see beautiful circle of expression on the surface, whereas on the other side, where you have the mutant, it doesn't get as much to the surface, which accounts for the reduction in the sodium current. It also results in redistribution of the protein and allowing it to interact with other things that will help to internalize the sodium channel from the membrane to bring it inside. And that gave us this general model where we begin to believe that CRIM2, through a series of interactions with other proteins, and upon simulation, is able to result in the endocytosis of the channel. Therefore, we have less channel on the membrane and less current. Okay, so good so far, but can we do this in vivo? So what did we do? We used CRISPR-Cas to make a mouse. We made a transgenic mouse, knock-in mouse with a homozygous 374A, so sumo-null. And so we asked, well, can this mouse have simulation of CRIM2? We did this in a couple of ways. One way shown here is proximity ligation. I'm sure you know what this is, interaction of proteins in the cell with two antibodies, and then the antibodies can be revealed through proximity within 30 nanometers to another secondary antibody. And the projection is bad, but there's red dots in here, but there's no red dots here, showing that the interaction is gone in these sumo-null mice. Most excitingly, if you look at these two, and this panel up here, A and C, what's showing you is that we get nice, beautiful sodium currents, 1.7, from wild type, but in the sumo-null, they're gone. Amazing. So now we've recapitulated our in vitro findings in an actual animal. And it's because of trafficking, so loss of trafficking. So the mechanism is starting to build. These animals otherwise are normally fine. They do have thermal analgesia. So if you put them on a hot plate, they can withstand the temperature a little bit more. And this is very reminiscent of the John Wood's Nav1.7 knockout mouse that he made many years ago. So we're in the same pathway for regulating these pain-like behaviors. We also looked at a lot of other behaviors in these animals. They don't have any response differences between wild type and the sumonull in the formalin model, in the acetic acid-induced writhing model, or the incisional surgical cut model. But amazingly, and this to me was probably one of the highlights of the results that we got in Tucson, when my lab was in Tucson, was this experiment. We're doing a spare nerve injury by cutting the tibial and peroneal nerve. The sural nerve remains intact, and we can look for areas of hypersensitivity in the animal with one prey, as you guys do. And amazingly, if you look at both females and males, of course, the wild-type animals in both sexes develop a lot of pain throughout the course of the experiment, but the CRIM2 sumo-null mice first did not develop the full extent of pain, and then they quickly recovered. And if we take these animals out to months and months and months, they have no pain whatsoever. So simply mutating one amino acid in this protein is able to prevent the development of chronic pain, which is pretty amazing. Remember, so sumolation is an enzymatic addition of sumo, and it's catalyzed by UBC9. But it can also be desumolated. So in a project awarded to Kimberly, she's on the audience listening in, so when she was in my lab, we got an NIH grant supplement award to study the idea that if we just boost the level of this protease, maybe we could shift it to this non-pain again. And we exactly did that. Using a dead CRISPR, we locked on to SENP to drive its overexpression, and guess what? Again, these animals had pain reduction. So these are experiments done by Kimberly, and it shows you that in this model of SNL, these animals that expressed through virus, the overexpressing through that CRISPR, this protease to drive it this way, they had less pain. So closing the loop on the idea that CRIM2 regulation is really sitting at the heart of driving the regulation of 1.7. Or CRIM2 stimulation is at the heart of driving 1.7. All right, with all of that, and that's now already been like 6-7 years, we had a good understanding of what was happening. So we thought, okay, can we do this using a small molecule? And at this point, I formed a company in Tucson with all the backstory I told you, a company called Regulonics, regulators of ion channels, merged them together, Regulonics, and got in some big heavy hitters. I formed the company with my wife, a wonderful physician scientist. I'm sure you know Gerald and Frank, and they're still on the board of this company. And we are moving some of these compounds I'll show you to hopefully to the clinical doorstep very soon. All right, so everybody's doing this. All the competitors, all the drug pharma companies are doing this. They want to block the channel. Should work theoretically, but it has not succeeded. Side effects, autonomic dysfunction, hypotension, and you need pain to live, so not a good thing. Our approach is regulation. So we are doing the stuff on the right. The idea is to understand the conversation the channel is having with auxiliary proteins, in this case CRIM2, and it's stimulated CRIM2. And if we can figure out a way to computationally find molecules to block the stimulation of CRIM2, we could revert it to the no pain stage. And we did that with the idea that UBC9, the enzyme that stimulates CRIM2, could be that target. With my wife's group, we embarked on an in silico docking campaign. We docked compounds on that grid around the lysine 374 and found two sites that are important. From this, one compound emerged called 194. 194 will drop down in the crevice between SUMO, UBC9, and CRIM2. And you can see the compound in this molecular dynamic simulation is pretty stable. It's sampling the space, but it's not allowing the simulation to come and be added on to CRIM2. So we use this compound as our lead compound to go forward. Through various methodologies, including some biophysical techniques, we found it only binds to CRIM2, doesn't bind to CRIM5, for example, does not bind to UBC9, very important because UBC9 is embryonic lethal, so we don't want to touch it whatsoever. So that's good. It inhibits the simulation of CRIM2. This was an acid developed by a former postdoc who's now a faculty Obama in St. Louis. And through microscale thermophoresis, we're able to show the reduction of the interaction between CRIM2 and UBC9. So the compound was working as it's supposed to work. But does it do what we want it to do, which is to regulate the channel? We don't care about CRIM2. Well, we do, but ultimately our goal is the channel, right? So that's the idea. So this was the most important experiment done, I think, around this question. And here's really the money shot. So on the top left, you see recordings from neurons, rat neurons, in presence of DMSO, nice robust currents. In the presence of our compound 194, which blocks simulation of CRIM2, we see a nice reduction, about 40%. But we want to make sure it's 1.7. So how do we do that? Well, we can use Protox. Remember, Protox blocks 1.7. So if we add Protox by itself, you see, again, a reduction from black to green. And if you add them together, this is in brown, we don't see any further reduction. So all of the reduction that we're seeing in the summary data from black to brown, black to blue, is because of 1.7 regulation by 194. So that was pretty amazing. What was even more amazing was it was super selective. It doesn't hit any of the other channels, the sodium channels. It doesn't touch the calcium channel. So that's exciting. It transcends across species. It inhibits currents in mouse, in pig, in human DRGs. We've since tested in dogs. It also works as well. And importantly, because we had made that mouse many years ago, we now had that mouse for the ultimate proof, right? So these mice have lower amounts of sodium currents. Yes, they do. But because this mouse doesn't have the simulation of CRIM2, when we give them the compound, nothing should happen. And nothing happened. So this was amazing proof that this is all happening through simulation of CRIM2 to regulate 1.7. All right. It actually works in other cells as well. So for example, trigeminal ganglia, this is Santiago Loya Lopez and Aida Calderon Rivera. These folks, these are postdoctoral fellows and now assistant scientists. And Santiago, along with Kimberly and Aida, recorded from trigeminal ganglion neurons and in the presence of a CRIM2 siRNA. And you can see, again, the currents are reduced. If you add 194, nothing happens. So again, it's CRIM2 dependent and CRIM2 simulation dependent. Paz, in the audience, hi Paz, also did this experiment where she looked at proximity ligation assay. You can see some beautiful red puncta here, but not so much here in the presence of 194, showing that there is real interference of the simulation or the interaction between 1.7 and CRIM2 because of 194. And you can see this in different ways. Here we have two other examples. We perform biotinylation to see whether the channel will get to the surface. It does not in the presence of 194 compared to wild type. And also using confocal, in the wild type, in the sort of the control, in the vehicle control, it gets to the surface, but in the 194, it does not. With the idea, again, registering that it's because of trafficking that leads to the reduction in sodium currents. And again, this really kind of promotes its redistribution of CRIM2 to other proteins so that the channel can be brought in. So it recruits endocytosis-related proteins to be brought in. So all that was sort of in vitro. What about in vivo? So this was the first experiment. We were trying to pitch this to, I think it was AbbVie or Abbott at the time in Tucson, and they asked us, do you have any in vivo evidence of this engagement of this mechanism? So here's the experiment. So we gave animals intrathecally 194 and killed them at I think about 2 hours after. And we purified CRIM2 and looked at its whether it's simulated. You can already see it's decreased. So this is in vivo, all in vivo. And then we also did the same thing in the spinal cord to see if the channel trafficking was affected. So Obama prepared spinal cord non-post density fractions, and you can see again, if you compare vehicle to the 194, there's a clear difference. So it's indeed evidence that in vivo we see suppression of stimulation of CRIM2, but also effect on trafficking of 1.7. So that was pretty awesome. And then, of course, we chronicled this through all of our pain studies. So one of this is the first one we tried. This is the spared nerve injury model. Again, cartoons up here, just to remind you over and over again. Here's the scenario. CRIM exists in a sumo state. UBC9 brings sumo to CRIM2, and this is what's sort of facilitating pain. And our compound interferes with this interaction, prevents stimulation, and then you have less pain. That's the idea, right? So just keep that in mind. So when we take the SNI animals, after about 15 days, they develop pain, as I'm sure you do this model here as well. And at this point, we wanted to figure out where the site of action of 194 was. So we gave the compound two different ways. Either we gave it in these animals in the paw, or we gave it intrathecally. And only when we gave it intrathecally did we see a pain-relieving effect. And combined with our spinal cord electrophysiology data, which shows that there's a clear reduction in excitatory postsynaptic currents, as well as in action potentials, we believe that this supports a central mechanism of action of 194. We hope to make it peripheral at some point, but I'll get to that in a little bit later. But this is sort of what we believe right now. Ultimately, we want to develop this as an oral compound, as a pill that we can take. And here we have another experiment where we did spared nerve ligation, and we gave animals, once they had established level of chronic pain, different doses of our compound. And you can see a fair degree of relief over a few hours, maybe 4 hours, but it's not 100%. And this is one of the reasons why we're still trying to optimize 194. I have a lot more data on this, but I will suffice it to say that it also works in chemotherapy-induced peripheral neuropathy. I'll show you that in the next slide. It works in formalin model, post-surgical model, model of itch, model of chronic constriction injury, as well as a couple of other models that we recently have been doing in New York and now in Florida. And this is the model of arthritic pain, as well as a very, very difficult pain, excruciating pain in human model, which is the trigeminal neuropathic pain. This is something we're really gotten interested in the last year or so, because there's a trigeminal facial pain foundation based out of Gainesville that I will also talk about a little bit later. Okay, as I said, I've always wanted to collaborate, so I send the compound to many different labs across the US and also to Europe. And here in the lab, Daniela Salvamini at SLU, what they did was a model of oxaliplatin-induced chemotherapy neuropathy. They did the experiment as you and I would normally do, which is to take animals, inject oxaliplatin, make sure they have the neuropathic pain, and give the drug. So to test reversal, right? That's what's shown in panel C. You can see the animals develop pain, and at this point, they give the animals 194, and it reverses. Beautiful. Reverses for about the same time we saw, so it all works. But either accidentally or serendipitously, they also did the experiment on the right, which is they gave the compound, 194, before they gave oxaliplatin, with the idea to test whether this could be a disease-modifying compound. And guess what? It is. So in here is the experiment shown. So the animals' weights are all the same, but in this case, when you give, if you focus your attention on the oxaliplatin plus vehicle, you see a nice development of pain. Vehicle is this gray bars, circles. They have no pain. But amazingly, when you give 194 before you give oxaliplatin, the animals never develop neuropathic pain. This is pretty amazing, because to me this is either it's a disease-modifying effect, or it's interacting somehow with oxaliplatin. It doesn't seem to do anything to oxaliplatin's ability to kill. We've tested that already. So it looks like it's maybe a disease-modifying effect. So that's pretty exciting. Opioids have a stronghold across the globe. I don't think ever we're going to have a case where opioids will be completely abolished, even with the increased scrutiny that's happening in the US and has happened. It's getting better and better, but it's still highly used. But with that in mind, what if we could take a very small amount of opioids that is non-toxic, it doesn't cause any side effects, and add 194. Well, with that idea, we do these synergistic studies, and here the bottom line is that if you combine 194 at a subtherapeutic dose with either gabapentin on the left or morphine on the right, you now see a a synergistic effect, so that now you see a much higher degree of relief where you saw nothing with the individual compounds at a very low dose. So again, ultimately, if our drug fails to be really good, we could combine it with either morphine or gabapentin to make it much more effective, but dial down the side effects of those two compounds. All right, so of course we've been looking to test side effects of this drug because it looks pretty good so far. So what about some of the side effects? So one of the big things with any kind of chronic pain drug, you're not going to take it once, unfortunately. You're going to take it many, many, many times, right? Over the course of a lifetime, that can be 25, 30 years or more. So does it lose efficacy? So do this, you can do a tolerance experiment where what we've done here is injure the animals with an SNL surgery and then implant these Alzette pumps that deliver a constant amount of compound over a amount of time. So this pump lasted about 14 days. And so here the pump comes on and it comes off. And you can see the animals that get vehicle, they remain in pain throughout the course of the experiment. The animals, and shown in the green squares, have the 194 added into the pump. And you can see as soon as the pump comes on, they have almost complete pain relief all the way until about day 10, day 12. And at day 15, or even, it's still pretty high, very significant. And even after the pump wears off, or it's stopped and there's no more drug, you still see a little bit of effect. So there's really no tolerance building from the repeated administration of this compound. Also with Todd Vandra's group and Andrea Homan's group in IU Bloomington and University of Arizona College of Medicine, we looked to see whether there was any addictive properties. The compound has no preference in the conditional place efferents on the left, and it doesn't also cause any physical dependence. You can test this by giving the animals naloxone, and then you withdraw the naloxone, and what happens is the animals will jump this naloxone challenge. So the compound doesn't do anything to that as well. So there's no physical dependence because of 194 either. We also looked at many other functions, including locomotor, not affected, exploration, not affected, anxiety was not affected, despair was not affected, olfaction. This is important because 1.7, the people that have this mutation that causes them to have no pain, these humans, they have no smell. They cannot smell. So that's a bad thing. I could develop 194 as the best drug, but if you lose your sense of smell, oh, that's not good. That's going to lead to a lot of problems, eating being the least of them. So that's not good. So that's a good thing that this doesn't do anything. And the last issue, the one hurdle that has been insurmountable by all pharma, everybody has tried, everybody's failed, is this idea of what I told you about earlier, which is the sympathetic engagement, the sympathetic autonomic dysfunction. So this is a compound called GDC-0276 from, I forget the company, but this compound and the compound shown on the right, they have this serious effect that even though they're safe drugs to take, clinically safe, and given to volunteers, the volunteers pass out because they increase the heart rate, drop the heart rate in these patients. Amazingly, our compound doesn't do any of this. So I'm pretty excited. And the reason, I'm sure somebody may ask, the reason for this is because there's cell-specific regulation of 1.7. And in the sympathetic ganglia, this is not likely happening. And I can give you another example of why this may be happening. So for example, we looked at nodose ganglia. So I had Santiago look at nodose. So 1.7 also controls the cough reflex. So you don't want to be targeting anything in the nodose. So we looked at nodose ganglion neurons. We're able to see beautiful sodium currents. When we applied 194, we saw no effect. We're like, what the heck's going on? Because if it's not a conserved mechanism. It's like, okay. Until we realized that one of the key proteins that helped to nucleate that complex that drove the endocytosis of 1.7 through CRIM2 was missing. And this protein NUM was the protein that was missing, or at lower amounts compared to DRGs, where we do see effect of 194. When we overexpress NUM in this system, so we took regular cells and overexpressed NUM, we see beautiful currents. But now when we put 194, we see the inhibition. So it's that missing piece. So this shows you cell-specific regulation, which also tells you that this is probably why we don't see the sympathetic effects. All right, I'll move along a little bit faster now. So the compound has come along famously well. It's still my baby. I love 194. It's my compound, but it's nothing compared to. I use the analogy of iPhone 0 versus iPhone whatever. What's it now? 17. So this is iPhone 0. This is iPhone, I don't know, 100. We have a compound now. So for example, the IC50 was 1.2 micromolar. The best compounds that have been developed in collaboration with NCATs are in the low nanomolar. And all the other things have been amazingly accelerated. It's much safer, it's better, no hERG activity. The half-life is far superior, as you can see, going from about 2 hours to about 22 hours. So it's super amazing. And these compounds are now being sold to a company I cannot disclose to be developed for humans. So I'm pretty excited about that. Okay, last few slides I'll tell you about is, as we move this, as you know, pain is very difficult to solve. Drug development for pain is filled with failure after failure. 99% of drugs fail in this space. So perhaps we can design some drugs for our pets. So a good friend of mine who lives in the neighborhood, she's a vet, she works on horses and small animals, and she came to me with one of her friends who has a cat with burn-related neuropathic pain. The cat's named Sandwich. So Sandwich tried basically, you can't see the needles, but there's a needle sticking out, so acupuncture. They tried all kinds of other things, compressive clothing, nothing worked. Gabapentin worked, but the Sandwich was basically sleeping. It just put the animal to sleep, no locomotion, nothing. So the owner was not happy. Ludo came to me, I gave her 194. She compounded it with her pharmacy. You can do that for non-humans. And she gave it to Sandwich. And lo and behold, Sandwich is all happy and living her best life. And so another example of showing you that something we discovered, in 2007 when I was a postdoc in Toronto, made my own lab, moved to Indianapolis, discovered the mechanism, went to Arizona, developed the company, made this compound, and finally we have something that can be used in cats and dogs. It also works for dogs. And we are now trying to do this in horses because they have this trigeminal neuralgia, which is this shaking phenomena that they get. It's not just them trying to ward off flies or something. It's a serious neurological condition. And sometimes they have to put the horses down for this. And Gainesville is very well known for thoroughbreds and horses. And so this is a big focus for the lab in the coming years. All right, one last thing I'll tell you before I quit is that what bothered me, I couldn't sleep at night, was not just because of my little kids, because of the idea of why the hell is CRIM2 only talking to 1.7 but not the other 8 isoforms? So this is something that bothered us for a long time. So Kimberly, in one of her beautiful papers, along with Harrison Stadden, one of my last PhD students from Tucson, came up with this beautiful paper where they together identified a unique region, this little piece here, in the channel that was important for all the interaction. This piece is not found in the other channels, and I don't think I have enough time to go through all of that, but I'll show you that. So it's basically in this first loop, this CRIM2 regulatory sequence. If you look in the other channels, it's somewhat there, but not the entirety of it. And we did some microscale thermophoresis to look at what binds best, and the what bound best was the piece from 1.7. All the others were were much more inferior. And then using a very clever trick that Stephen Waxman's lab had first brought out, we put a halo tag on the beta 4, which is concatenated with the channel, so we could look at surface exposed channels, look at the channels that we transfected. And then what we did essentially is from the wild-type 1.7 channel, we just flipped in the other pieces. So basically, we just made this either 1.1, 1.2, 1.3, 1.4, and so forth. And when we did that, basically, we lost all the current. So all of that regulation was coming from that little piece in 1.7 and nothing else. So this is amazing. We made this into a peptide with our usual peptide-based tricks and added into cells, and we can also see a similar regulation. We are pretty excited about this because it also inhibits excitability, but the real excitement came from putting this into, I think that's on the next slide. So we tested this in animals, but in the next slide, on panel C and D, we put this little peptide, this time from the channel, remember. It's not from CRIM2, it's from the channel, this little 15 amino acid or 12 amino acid piece. And when we did this and put it in a virus and put it in animals that had SNI, now you see pain relief lasting for weeks and weeks and weeks. So this is at day 38 here for males and females. So this could be potentially be a gene therapy approach. This has been tried. Gene therapy for 1.7, I think, could be a viable way to come up with a chronic solution for a chronic problem, which is chronic pain, right? So this is something we're pretty excited about. To close the loop, Eric, he's in the lab now. He's probably also on Zoom. Hi, Eric. Eric, and we decided to make this mouse. So we made a mouse that's lacking this little piece. Anybody want to guess what happened to this mouse? Are the currents going to be bigger or smaller? I hear smaller. You are right. Absolutely smaller. Look at that. Amazing. Proof that the mechanism works. It's amazing. Absolutely amazing. And then Eric did the most important experiment in vivo. The animals, naive animals, they have no change in their sensitivity to pain. But guess what? When he did an SNI injury on these animals, spared nerve injury, the wild-type animals, of course, develop pain. The SNI animals for this Nav1.7 delta CRS, they, again, do not develop and then start to recover. So pretty excited about closing the loop on this mechanism this way. All right, so I've told you a lot about, this is my last slide. We began with an idea that we want to regulate the channel, not block it. Because blocking had been a problem by pharma and all the, across the globe. So we came up with this protein called CRIM2, discovered that it could really regulate the channel. It could lead to a discoupling. If you uncouple the channel from CRIM2, you could see some real nice effects on current, surface trafficking, which led to decreased excitability and then loss of behaviors. And we verified this through in vivo modeling, but also made a genetic knockout to confirm this. So this is pretty exciting. On the other side, we also use SENP to drive this, and we probably will look for SENP activators, which is kind of a hard thing to do because they're more, it's kind of a more common kind of enzyme. So, and we also, along the course of doing these kind of things, also have embarked on some sophisticated experiments that Paz contributed to while we were in New York. You're looking at these live cell imaging going in and out. These are single molecule clusters of channels. So this is looking at a protein that's been conjugated to the channel, and we can do, because look at the resolution. This is 1 micrometer. So this is basically looking at the entirety of either one channel or a couple of channels. So we're now looking to move this technology into more kind of getting a real understanding of what's happening with some of these molecules and these channels in a real life or lifetime tracking. But I think this method of regulation of channel could be a very tractable approach as an alternative to developing pain drugs for against this channel. And of course, the most important slide, I presented this yesterday, got some laughs. Making Mexico great again. And these are the wonderful people. Kimberly's up on the left, very small here. This is Paz, Santiago Lopez, Eric, and then Aida, Calderon. And this is the party we had for Kim because she just got a position in Texas A&M. So we're very, very excited for her to start her own lab. We'll still continue to work with you, Kim. And this is a collaboration with my wife's group. And I do these for these 3 monkeys. And happy to take any questions. Thank you very much. Oh, and also before you guys, while you think about that, I will do what I do always, which is take a selfie. I'm not good at it, but still do it. I'll do two of them. Thank you for hearing me. Questions?
¿Alguien quiere hacer alguna pregunta? ¿Sí? A ver, tenemos a David, luego.
Thank you very much.
Thank you.
I just wonder about the 194 compound, if you have some sort of kinetic data, absorption, distribution.
Yeah, we do. Thank you for the question. So the question is about pharmacokinetic data for 194. So 194 is my baby. I love it. It's not the best compound. It has some liabilities on this. It is heavily metabolized, quickly metabolized. The half-life is less than 2 hours. It doesn't have very high accumulation in the periphery or the brain, and it basically because it's being metabolized very quickly. And we found this out from a very unfortunate series of 3 long full-day experiments on horses. So we had 4 thoroughbreds who we got permission to give naive animals, no pain. We gave intranasally these big horses. They're about 600-700 pounds. We gave them a lot of compound, and we saw we did blood sampling and urine sampling for 24 hours, and we see nothing even after 5 minutes. In the first 5 minutes, it's gone already. So kinetically, and the ADME is really bad. But I'm happy to report that because of our collaboration with NCATS at the NIH, that issue has been solved, and the compound is beautiful. Our 2 lead compounds are absolutely gorgeous. One is restricted to the periphery, and one is both central and peripheral. So we have just sold those to a company I cannot tell you about because I'm not allowed to, but they're going to take that peripheral compound and start to do IND enabling and to move it into humans. Yeah, thank you.
Here.
Other questions?
Thank you for your presentation. Pretty much the same question.
Yeah, thank you very much. So the question is about toxicology. We have indeed done toxicological work. So the toxicology we did was to inject increasing amounts, so the maximum, to find the maximum tolerated dose, which is ridiculously high, greater than 300-400 milligram per kilogram. And what we did was to give the compound for 10 days repeatedly to animals. And then at the end of the 10th day, we haven't done the 28-day tox yet. After 10 days, we sacrificed the animal and looked at all tissues that we can lay our hands on, and using blinded approach, of course, with a pathologist. And there's nothing different between the control vehicle treated or 194. So at least at the level of histopathology, after a 10-day massive dose, and I mean massive, probably 100 times the ED50, the dose for efficacy, we see absolutely nothing. And you saw a little bit of what I showed you when I was telling you about the side effects. We don't see any neurotoxicity, any kind of sedation. So we've done those kind of evaluations and histopathology, but that's about it.
And another question, probably not strictly related to pain, but to Nav1.7. In the lab, the Montserrat Fuentes found that probably the binding of cannabidiol to Nav1.7 accounts for the not synergistic effect of phentoin and cannabidiol. So my question is quite big. Any insights on how Nav1.7 might be related to other disorders, probably related to aberrant plasticity as pain, as epilepsy?
That's a beautiful question. I love that. I'm aware of that paper with the cannabidiol and the phenytoin. You know, 1.7, so all of us thought 1.7 is a neuronal channel, right? Makes sense. Drives active potential, so must be neurons. Two things that have come out recently, one big paper that came out recently that has challenged that dogma. A paper from Dr. Liu and Waxman in Nature of this year, or 24, I think, showed that 1.7 is present in chondrocytes. What the hell is it doing in chondrocytes? These are not excitable cells. So what they showed was in about 17% of chondrocytes, there is 1.7. And they mapped out a mechanism as to what it's doing. And what they showed essentially was if you delete 1.7 from the DRG, as John Wood did and many others have done, you have pain reduction, right? But if you delete 1.7 only from the chondrocytes using some special promoter, you not only have pain relief, but the development of things like osteoarthritis are gone. I mean, exactly what your expression of what? That's exactly what I read the paper. I saw the abstract, like, what the hell is this? This has got to be a mistake. It's crazy. So you're absolutely right. I firmly believe that 1.7 is likely to be important in other unknown areas, which is what I said in the beginning, that we don't know fully well the biology of 1.7. We haven't come up with a drug for something that we've known for about 30 years. We have mutations, we have beautiful compounds. Why don't we have a drug that works, right? So it's because of things like that, the autonomic dysfunction, the working in chondrocytes that came out of nowhere. So I imagine there may be other expression sites, and Ted Price's work on transcriptomics of many different humans with pain, without pain, different diseases, is starting to reveal that there may be changes in 1.7 as well. So yes, I think the answer to your question will need to be to wait until we really investigate this in a much more thorough fashion. But I agree with you. I think that it will be important for elements of plasticity. John Wood's group with Sasha Alice a few years ago showed that 1.7 is also present in the postsynaptic side. So it can regulate the postsynaptic coupling as well, which is very rare. You would never have thought of that. And so I think the story is not finished yet. Yeah.
Hi, Dr. Khanna. Do you know if in the market or in any other labs there has been drugs like yours worked on, like even for other illnesses or channels?
Yes, absolutely. So regulation is, I wish I could claim I discovered it. No, I discovered it from CRIM2, absolutely, and my lab did it. But gabapentin, what do you think it works on? It works on the alpha to delta. That's a trafficking chaperone. It's on the outside. Ours is on the inside. So absolutely, there's great precedence for regulators of ion channels. And in fact, the idea is pretty nice, right? You don't want to block your target protein. You're doing something to the protein-protein interaction. One advantage of that, as we see in our case, is that the protein interaction does not exist in the sympathetic ganglia, which is why we don't see that side effect that everybody else sees. So it's actually not a bad idea to go after protein-protein interactions of channels, receptors, kinases, whatever. Problem is, they're very difficult to do, right? But yes, there are beautiful examples. Gabapentin is one. Gabapentin has other effects on other things as well. But clearly, it started off, or its claim to fame, Annette Dolphin's group showed many years ago that it totally affects, you guys have done a lot of work on gabapentin. Gerald's done a lot of work on gabapentin, showing that it affects the trafficking, and this is how it starts to work. So absolutely, that is a precedence for it, and hopefully this will be, for the 1.7, this is a novelty. So hopefully this will work.
Thanks, Professor, for your talk. What about the difference between the current density between peptidergic and not peptidergic neurons? And what about with the voltage threshold for fighting action potential in that neurons?
Yeah, that's a great point. So the question is whether we've labeled cells with, for example, IB4. And so we're starting to do that now. I think Ida already left, but those are experiments she's doing now. I don't expect it to be different. I think, as you can see, 1.7 is expressed in a lot of different cell types. So I don't expect that. You saw from one of our data sets that we did affect the evoked release of CGRP. So likely it's going to be there as well. I do not know the answer to your second question without whether the thresholds are different. A lot of people, including Cheryl Stuckey and others, have shown that the DRG neurons are a heterogeneous mixture, right? There are all kinds. And now, we used to think of it historically as small, medium, large. Now, TED and other groups have shown that there are like 17, 18 different kinds of neurons transcriptionally. So we don't really actually know what's happening with those. So I think that is something that could contribute to the efficacy of our compound, but my prediction is that it's going to affect both. So I don't think it's going to discriminate between them. Yeah. Thank you. On the outside.
Thanks, Doctor. I was wondering or thinking in the Nav channels. It's well known in the scientific literature that Nav channels are differentially expressed between males and females. And how do you explain that we can't see any difference on the effect of these drugs between sexes?
Thank you for that question. So sex differences in pain have been reported for many years now. So the answer is, I didn't show you that data.
Okay.
So if you, in the paper that we described, the reporting of the mouse, we see sex differences in a model of veratridine. So veratridine is a sodium channel opener. So if you open the sodium channels too long, it can cause pain in an animal. So we took animals, gave them veratridine, and male CRIM2 null animals had less pain than female CRIM2 null. And in that paper, we also reported something that nobody reports. We also separated our recordings by male and female. Most people just clump all the recordings together, right? So we differentiated between male and female, and we see a clear difference. So in females, we see a much more pronounced reduction because of loss of simulation of CRIM2, but males we don't. And for this, until today, that was probably 7, 8 years ago, we have no idea why. We tried all kinds of hormones, supplementing them, changing them. We tried TNF alpha in a nice paper with Gerald Zamponi's lab, and we don't know what it is. But yes, there is at the in vivo level some sex differences. With 194, we don't see any sex differences. Don't know why, but again, we only saw that sex difference in that one behavior, the veratridine-based behavior. So with the SNI, we didn't see anything. So my hope would be that there will be no sex difference, because we don't want to develop a drug for only men or only women. That would be kind of bad. So I don't know why there's no sex differences, and that's something we haven't revisited. But that's a great question. Thank you.
Thanks.
Oh, I love it. You gotta take a picture. I have to take a picture with this. Somebody take a picture. No, but with Benicia, with you. Okay. With the Cinevista in the back. Okay. Okay. 1, 2, 3. Thank you very much. Thank you all of you. Thank you. Gracias a todos.